Good morning, everyone. Great to be here in beautiful Rome, and looking forward to connecting throughout the conference. My name is Tamer Mohamed, CEO of Aspect Biosystems. Our company is based in Vancouver, Canada, and we're focused on creating what we call bioprinted tissue therapeutics that we believe will have a big impact on the way we address disease using cellular medicines of the future. So I don't believe I need to convince this crowd, but needless to say, at Aspect, we have a fundamental belief that cells have an incredible therapeutic potential to treat the underlying cause of disease. But we also believe that cells could only take us so far. They've demonstrated a significant amount of impact in terms of treating the underlying cause of disease. But we look at Aspect, we look at cells as the building blocks for creating higher ordered biological tissue therapeutics that we believe could treat some of the most elusive diseases. And so what are bioprinted tissue therapeutics? So these are cellular medicines that we build using our 3D printing technology. We incorporate cells with other biomaterials, and we implant these tissues into the body to replace, repair, or supplement biological functions inside of the body. And so our goal at Aspect isn't necessarily to create an entire organ with all of the intricacies and complexities of an organ, but rather our focus is to identify organ functions that have been lost due to disease, then harness the power of cells that could address that particular mechanism that has been lost, marry those cells with our technology, deliver those cells into the body, but not just keep them there and deliver them there, but provide them with the best place for sustained function and ultimately efficacy. So a prime example of this is our focus in type 1 diabetes, where our goal is, of course, to replace those islets that have been lost due to disease, and providing those islets with the best chance of success, the best environment inside of the body to replace that missing function that has been lost due to that autoimmune disease. But other examples in the metabolic space include liver, a big area of focus for us, and an example of that is urea cycle disorder, where again we're looking to replace one particular function that has been lost in that organ that is responsible for over 50 functions, so that we could avoid, for example, the need for liver transplantation, and rather replace that one missing function that has been lost. In this case, that function is metabolizing blood ammonia and restoring urea cycle. So all of the indications that we choose are indications that we like to call thermostat indications, where we could tell if the disease is working simply through a readout from a blood biomarker. In the case of type 1 diabetes, that's of course blood glucose, and of course for other indications we would look at other markers. So by way of introduction into the company, Aspect was spun out of the University of British Columbia in Vancouver, Canada. This was based off of research I was conducting with my co-founders. We left campus in 2017 and have been making significant progress since then. We've raised over $150 million in funding. We have a team of over 100 scientists, engineers, and therapeutic developers that are committed on this mission to deliver high impact to patients using our technology. We have a broadly applicable platform technology, but we are also very laser focused on our pipeline, which is heavily focused on metabolic and endocrine indications to start off with. We have vertically integrated operations in Vancouver, Canada, with a 50,000 square foot facility that brings together our translational research, our engineering, and we're currently focused on our GMP manufacturing that we are setting up for initiation of clinical trials over the next couple of years. And lastly, we have a broadly applicable platform, so we've also been focused on partnering in specific indications, and we're very excited to have Novo Nordisk as a partner in diabetes and obesity. So our platform technology really we see as industry leading and really built as a full stack platform. And what I mean by that is it allows us to go all the way from the design of these tissues to the manufacturing and ultimately the implantation to facilitate a functional cure. So the process really starts in a computational setting where we are designing these rationally designed bioprinted tissue therapeutics. Then we move into the real world where we're taking stem cells, incorporating those with multiple different materials that have different functions, including providing the best environment for the cells to live, encouraging vascularization in the body, providing a level of immune protection, providing durable materials to facilitate the implantation and retrieval of our tissues. So all of those biomaterials and the cells are brought into our microfluidic bioprinting, which allows us to very quickly, rapidly combine all of these materials and manufacture in a single step our cell-based combination product. So a human-sized tissue could be manufactured in the matter of minutes, and these tissues are then ultimately implanted into the body to facilitate efficacy and replacement of a function that has been lost due to disease. So just to provide a close-up view or representation of at least the bioprinting parts of the process, what you can see here is a close-up of our microfluidic print head. You could see different channels with different materials that have been labeled with different colors. So these would be essentially the different input materials that are brought into the printing technology in liquid form. We then introduce this yellow material that surrounds the cell-loaded material and cross-links it instantly into a solid fiber about the thickness of a human hair. This fiber is then patterned into whatever structure that we've designed in software, and we could again very quickly create these structures. This is just a representative structure where we could create this multi-layered structure and then take that and ultimately process it or implant it into the body. So taking a closer look on our platform, really what we've enabled is an unprecedented level of control over the overall tissue structure and ultimately the function. So on the left-hand side is a visual representation of one of these multi-layered tissue structures. At the macro level, we have significant control over the overall tissue structure, but very importantly, at the micro level, we have a lot of control, and that's where a lot of the magic really happens. So this is one example of one of these microstructures where we've loaded cells in what we're calling the core, and so we could use a biomaterial that brings out the best functionality for that particular cell type. So in this example, you could see islets loaded in the core and functional as demonstrated by dysosome staining, or these cells are essentially insulin positive. Then we introduce a thin shell, and this is a totally different material. So this is a hydrogel material that is semi-permeable and allows what we want to get into our tissue to get in, what we want to get out. It allows those materials to get out, but importantly blocks any cell-mediated attack. And so when we look at a cross-section of that tissue, we could see that we don't have any immune cell infiltration. And finally, we introduce a outer material that is built to integrate and to interact with the native tissue inside of the body. And so this again is where we're looking to encourage vascularization, dampen any foreign body response, and ultimately achieve that harmony that we need between our bioprinted tissue therapeutic and the native tissue inside of the body. And so what we're able to do is we're able to bring in all of these different parameters into a single system and optimize for the particular problem while still keeping this as a single combination product that's fully retrievable. And so these are, of course, very sophisticated technologies and cellular therapies, and so that calls for very sophisticated manufacturing approaches to allow us to have a high degree of control over the overall process. And here's just a simple example demonstrating how we're able to achieve closed-loop, single-step manufacturing of these cell-based combination products. So what you can see on the left-hand side is a close-up view of the cells within the fiber as we're printing them using our microfluidic technology. So those lines on the outer side of what you're seeing here is the outer bounds of that fiber. And so this is a camera that's looking directly at that fiber. You could see the cells within the core. We're able to segment or do visual segmentation on that fiber. So that image in the middle there, that's not coloring in the fiber. That's our processing able to detect the different features. And we're able to do this throughout the print process. So at the end, you're able to get essentially a report over the overall key parameters and look at any deviations so that you could control the process, and you could also control that throughout the manufacturing step as well. And so switching gears from the platform to our pipeline, we're applying our platform technology towards metabolic and endocrine indications. Pancreas is a big part of our focus. That's specifically focused on using islets in the fields of type 1 diabetes, type 2, and obesity, and we're working on that with Novo Nordisk. Liver is a next big opportunity for us that we're very excited about. This is where we're looking at developing our liver tissue technology and bringing that towards the clinic across multiple areas, including liver failure, as well as hereditary liver diseases. And we also have multiple other endocrine indications that are earlier stages of development at the discovery stage. So I'll highlight our pancreas program as well as liver, starting off with pancreas in the field of type 1 diabetes. So type 1 diabetes, as everybody here is aware, a really debilitating disease. And it's one of those diseases that you only realize how debilitating it is the more you speak to patients. And this is what ultimately motivates us. And so right now the gold standard is insulin injections and pumps, which come with a lot of challenges and reduced overall life expectancy. This is a disease that's diagnosed at adolescence, and so these patients have to live for the rest of their lives with this challenge. And so our goal, quite simply, is to provide a islet replacement therapy without the need for chronic immune suppression. And the initial population that we're targeting is the one that suffers the most from this disease, and that's specifically hypoglycemia unawareness as a sub-patient population. And so what we've demonstrated to date is that we can achieve robust control over blood glucose in multiple different animal models, both immune deficient and immune competent, both in a xenogenic and an allogeneic context. And so what you can see here are several different sample readouts from our data sets. On the x-axis is all of these graphs is days post-implantation of our tissue, and on the y-axis is blood glucose, and these are all STZ-induced diabetic animals, and so we're able to very quickly bring the levels of blood glucose in these animals down to normal levels across these different animal models. And so last year, we were very excited to announce a precedent-setting partnership with Novo Nordisk. We're excited to have chosen them as a partner. And so in this partnership that's worth potentially more than $2.6 billion, we're focused on developing up to 4 different products in the fields of diabetes and obesity. We're starting with type 1 diabetes as an initial indication. And so Novo Nordisk, needless to say, brings significant expertise in the space, a rich history in this area, a commitment that we're aligned on to defeat these serious diseases that we're tackling. And also, beyond the economics and the rich history that Novo brings, they also bring significant expertise on the cell therapy side that we're leveraging. And so we're combining their stem cell technology, their manufacturing capability, and their development experience with our team and our technology to create game-changing therapies in the fields of diabetes and obesity. So switching gears to liver. So liver, again, is an area that we're very excited about. The liver is responsible for over 50 different functions. And in many liver diseases, only one or several of those functions are lost. But we're also in a situation where we have to take a brute force approach in solving these disease areas in the liver space by relying on organ transplantation. And so our goal is to avoid that and solve the particular malfunction by implanting a hepatic tissue that bridges to recovery or replaces that organ function that has been lost. So the way we look at liver is in 2 big areas. The first is acquired liver diseases, as well as secondarily genetic diseases or hereditary liver diseases. One of the applications that we're focused on is acute liver failure. This is where patients most often, unfortunately, don't make it through an acute episode. Their livers end up failing, and within a matter of weeks, if they don't get a liver transplantation, again, in most cases, they don't make it. So our goal is to provide a bridge to recovery for these patients so that they can get a liver transplantation or bridge to recovery. And so what we're showing here is in animal models of acute liver failure, we're able to bridge to survival in about 67%, or we are able to increase the survival rates by about 67% when we compare to the sham control. But not only acute liver failure is what we're focusing on. Ultimately, we're able to demonstrate that our liver tissues are able to address multiple clinically relevant functions that are also correlated to specific diseases, including ammonia detoxification, Phe metabolism, and other areas. And so again, our goal is to apply this liver tissue across multiple different liver diseases. Last but not least, I really want to highlight our team. It's always easy for me and very proud for me to stand up and share the great progress we're making, but back home in Vancouver, Canada, they're making all of the great progress and putting in all of the hard, long hours to make this potentially possible. So we have a phenomenal team with a lot of experience in filing INDs and bringing different advanced therapies into late stages of clinical evaluation, and we look forward to leveraging that experience in the years to come. And finally, to bring it all together, we're pioneering this new area that we call bioprinted tissue therapeutics, and to drive that forward, there are several key pillars that we're really bolstering, starting with our full stack platform that we see as industry leading. We're applying that to multiple areas in the metabolic and endocrine space. We're very selective and opportunistic in partnering with best-in-class partners, including Novo Nordisk, and then we're very excited about that partnership. And lastly, we're big believers of vertical integration to provide the speed and quality that we think we need to deliver to give us a shot at addressing patients with these debilitating diseases. And so we're building our GMP manufacturing and look out for updates to come in the next several quarters on that front. So with that, I would like to thank everybody, and I went over, apologies, and looking forward to taking any questions offline. Thank you very much.